Well, hello everyone. First of all, thanks to the organizing committee for letting me present this little work I have here. That is most of it part of my master's thesis, and this presentation, which, as you said, is entitled Morphological Alteration of Pyramidal Neurons from the Contralesional Hemisphere After Ischemic Stroke. So, as you might know, stroke is one of the major causes of death and disability worldwide. In fact, with more than 50 million cases every year, the world is facing like a modern epidemic. It is traditionally defined as a neurologic deficit arising from a focal and acute injury in the central nervous system, in which ischemic stroke represents the most prominent type, accounting for more than 80% of the total cases. The ischemic insult is basically explained by an artery occlusion that leads to blood and oxygen deprivation in a given brain area. Much emphasis has been put into understanding the insight of this focal lesion, which embrace an infant core and ischemic penumbra surrounding. However, it is worth noting that the patient undergo a long and frustrating rehabilitation process, which do not guarantee a total recovery from the lesion. In fact, interesting studies indicate that remote areas connected to the infant core are also affected as a consequence of a process known as diaschisis. One of these areas is the contralateral hemisphere, which is crucial in the patient recovery since it participates in the reorganization of neural circuits after the stroke event. The alteration of this contralateral hemisphere may be implicated in the suppression of functional recovery in the ischemic patient. Given this, the goal, as I said, of the present study was to analyze possible microanatomical alteration of pyramidal neurons in layer 3 from the contralesional somatosensory cortex in the ischemic stroke mice model TMCIO. This was assessed through two different approaches: the study of neuronal complexity regarding several morphological parameters of the apical and basal dendritic trees, and the study of dendritic spine morphology. Well, here I present an schematic workflow of the material and methods. We use the transient middle cerebral artery occlusion mice, for now on TMCIO, as our experimental model, in which we first perform an initial staining just to delimit the extension of the ischemic injury throughout the cerebral cortex, and then perform intracellular injection on individual pyramidal neurons in order to 3D reconstruct each of them using neurolucia software. As I mentioned, we use the TMCIO mice model in our work. Briefly, it is based on the occlusion of the middle cerebral artery using a nylon monofilament during a given time, in our case 1 hour, which results into the atrophy of most of the lateral side of the cerebral cortex. I like to point out that this surgery was performed at the Laboratory Experimental Stroke Research of the University of Munich. Along with the TMCIO animal model, we used the sham mice just as control animals. On these mice, we carried out the intracellular injections. We performed this over fixed coronal sections of tissue previously incubated with DAPI, which help us visualize the cellular nuclei, as you can see here, of the contralateral somatosensory neurons we want to inject, and actually injecting them a fluorescent marked lucifer yellow, which result into the individual marking of the pyramidal neuron and the dendritic spines. Then we took photos with confocal microscopy and 3D reconstruct each of these neurons and their dendritic spines using neurolucia software, as I mentioned before. Going on with the results, what we first observed was a clear and significant reduction in the apical dendritic tree complexity in TMCIO mice, regarding several morphological parameters such as dendritic length or the number of intersections, as you can see in these graphs, shown in blue TMCIO and in orange the sham mice. This was a study using a Scholl analysis that measured every morphological parameter as a function of the distance from the soma. However, these differences were not found for the basal dendritic tree complexity, in which for both groups, sham and TMCIO, it shared the same tendency for any parameter studies, as you can see in these graphs. So to further analyze this, we took a look at dendritic spine morphology. We measured dendritic spine volume and length through different kinds of studies, only finding significant changes in the frequency distribution analysis, both for apical and basal dendritic spines. All these alterations, although they are subtle, are quite robust, as you can see from the p-values extracted from these graphs, except for the apical dendritic spine volume. In conclusion, in the present work, we have shown that stroke reduces neuronal complexity of the apical dendritic tree in the contralesional hemisphere, and that also induces changes in the dendritic spine morphology in the contralesional hemisphere. All these changes could translate into a more than possible alteration of the neuronal circuits in the contralesional hemisphere after a stroke. This is of great importance, and given this result, it is a need to develop new therapy approaches that not only minimize the damage caused by the infant core, but also protect the areas that are connected to it, and which are, as I said, essential for the patient recovery of normal function. And well, that's all I have to say. Let me just quickly thank my mentors, Paula Merino Sarrais and Javier de Felipe, for the outstanding mentoring during this work, and Asta Kastauskaite for her help with the methodology, as well as the Experimental Stroke Research Lab for the generation of the model. And just mention that this work was part of a bigger European project known as MIST, in which we collaborate along other European labs. Well, now that's it. Thank you all. I'll be glad to answer any question you might have.